Gastrin. Gastrin is a hormone the stomach produces that stimulates the release of gastric acid. It is located in the stomach and upper small intestine. Gastrin allows the stomach to break down proteins swallowed, such as food, and break down certain vitamins.
DNA is made up by nucleotides, which consists of nitrogen bases, sugar phosphate, backbone, phosphoric acid, and is bonded together through hydrogen bonding. The DNA contains genetic instructions for the development and function of living things. DNA is located in the nucleus. The gastrin gene. The gastrin gene is located in chromosome 17. The gastrin is located in the part of Q21.2 and in the nucleus.
Transcription is a process of manufacturing RNA to DNA. There are 3 steps to this process. The first step, initiation, the DNA molecule unwinds and separates to form a small open complex. In step 2, elongation, RNA polymerase moves along the template strand, synthesizing on an mRNA molecule. In step 3, termination, the mRNA that is formed leaves the nucleus. mRNA encodes the amino acid sequence. rRNA associates with proteins to form ribosomes, which structurally support and catalyze protein synthesis. tRNA transports specific amino acids to the ribosomes for protein synthesis.
In RNA processing, the pre-mRNA goes through modification and splicing to become a mature mRNA, ready to leave the cell. A pre-mRNA consists of 3 exons and 2 introns. During modification, a cap and a tail is added. These work as protection during the transformation. In splicing, the 2 introns are removed. During translation, the mRNA is decoded to build a protein of amino acids. The process takes place in either the nucleus or the cytoplasm. The process is divided into 3 steps: initiation, elongation, and termination. In initiation, the ribosomes assembles around the mRNA's start codon. In elongation, tRNA transfers an amino acid to the tRNA corresponding to the next codon and continues to make an amino acid chain. Termination, we reach the stop codon. The ribosome releases the ready polypeptide. The levels of conformation is primary, secondary, tertiary, and quaternary structure. The primary is just an amino acid chain. The secondary is loops and coils of amino acid chains. 2 common structures is the alpha helix and the beta sheets. The tertiary is folded secondary structures. And the quaternary is multiple tertiary subunits bonded together.
Exocytosis. In the cell, the proteins that leave the cell are typically made up by the rough endoplasmic reticulum. The proteins are packaged by the Golgi bodies. The Golgi bodies and vesicles exit, and the Golgi bodies on preparation to leave the cell. The merged items travel through the cytoplasm and can exit through the cell membrane. That is the process of exocytosis.